And following this talk, there will be an opportunity to ask our speaker, Dr. Laura Fachal, questions. So please, as I always mention, submit your questions throughout the talk using the chat function. I know probably all of you will wait until the end, but again, I encourage you to do it while you hear Laura speaking. I will then collate the submitted questions and put them to Dr. Fachal on your behalf following her presentation. So now I am delighted to introduce to you our speaker, Dr. Laura Fachal, a veterinary medic by background, studied genetic epidemiology of radiation-induced toxicity during her PhD in the field of biotechnology. She came to the UK as a postdoc with a Marie Curie IF Fellowship to work at the University of Cambridge at the Center for Cancer Genetic Epidemiology. Dr. Fachal's work there in this Center for Cancer Genetic Epidemiology was to determine through fine mapping approaches how common genetic variants increase the risk of developing breast cancer and increase radiation-induced toxicity. Since then, she has become particularly interested in understanding the biological mechanisms by which genetic variation affects complex phenotypes. So her current postdoctoral role at the Wellcome Sanger Institute focuses on inflammatory bowel disease. She was awarded with the Young Researcher in Human Genetics Award by the Spanish Association of Human Genetics back in 2015. And she also has a long list of publications, among which we can see various Nature Genetics and Nature. So finally, it is also a great pleasure for me to introduce Dr. Fachal today, because we have known each other for a while, long before we've both become part of Lucy Cavendish. So over to you, Dr. Fachal. Enjoy your talk.
Thank you so much, Christina, and thank you so much, Lucy, for giving me the opportunity to be part of this Live from Lucy event. So let me share my screen. And okay, so today I'm going to be talking about generally about common variation in the DNA and how this might be influencing our risk of developing what we call common diseases. And I will be going through some examples with two diseases I've been working with. One is breast cancer, and currently I'm working with inflammatory bowel disease, which is an immune-mediated disease. And I'm currently based at the Wellcome Sanger Institute within the Carl Anderson's team. So I will start. Let me see, how can okay, I need to go there. Going back in time, 30 years, to when the Human Genome Project started. It's specifically relevant for me as well, because I'm based now at Wellcome Sanger, and Wellcome Sanger Institute was at the very beginning created in order to help to participate in this massive international project. Before the completion of Human Genome Project, we did know something about certain parts of the genome and which was the sequence, which was the letter code in that particular regions. But I like to think on this as a kind of puzzle. We have the different pieces, but we haven't put all these pieces together, and we didn't know which was the relationship between these pieces. So within the Human Genome Project, we managed to put all this together and then managed to get a sequence for each one of the chromosomes from one end to the other one. So as humans, we have 23 chromosomes, and we have two copies for each one of these. One is being inherited from our biological mother, and the other one from our biological father. And I like to think of them as kind of books within an encyclopedia. And with the Human Genome Project, we got like the first edition of this book. However, because of the technology at that time, we were not able to read all the different pages. There were some pages where because they were sometimes like the same word was repeated many times. We were not able to read them perfectly well. So we knew that there was a gap there. And in a subsequent edition of this encyclopedia, we managed to fill that gap. There were also some pieces that we didn't know where about to put, some of the pieces of I was showing before on the puzzle. So we were keeping them on site. And later on in subsequent editions, we managed to put them whether they were in chromosome 1 or chromosome 18. So this is still work in progress. And think about that, it's been like 30 years. There is a really interesting project these days where using the new technology, they are managing to get a complete sequence within the chromosome from end to end, but only one chromosome has been released so far, which is chromosome X. So what we learned from the Human Genome Project? We got an estimate of how many protein coding genes we have in the genome, which is about 20 and 25,000. And what are these genes? These genes are portions of the DNA that contains the information that the cells use to create the proteins, which are the bricks that makes us as a human beings. So if you think about the human genome as what we say in the chromosome as a recipe book, in this case, then when you the piece of DNA that has the information for a particular gene, it's a recipe within the book. So this is being copied into what we call messenger RNA, which would be like copying one of the recipes into a note. And then this is being translated into proteins, which would be making the cake from the cake recipes, from the note we made. Another thing we learned after the completion of the Human Genome Project is that our sequence is really, really similar. So, and another person and myself might have 99% of our sequence is the same. Every single position will have the same letter. However, there are specific positions in the DNA that can be what we call polymorphic. So there are more than one option available. So for instance, in this case, this specific position, there might be a C or an A. So maybe 50% of a group of 100 people will have a C in that position, and another 50% will have an A. And each one of these alternatives is with what we call alleles. And I'm showing here the most simple way of polymorphism, which is what we call single nucleotide polymorphisms. So polymorphisms are behind most of the human diversity we can observe, the physical human diversity, from whether we are able to curve our tongue in that way, to whether we have freckles or not, or which is the color of our eyes. But they are also behind whether we have dimples or not when it's smile, right? So going back to these polymorphisms and to the biology, I was mentioning before that we have two copies. We have one copy of our chromosome that is coming from our maternal biological mother, another one from our biological father. So that means that for each one of these positions in the population, there are three possibilities. For instance, for this example, we can have a G or a G, we can have a G or a C, or have two copies of the C allele. But for purposes in order to be more simple, in this case, I'm going to show all the images like if we were having only one copy. So there is only a possibility. We can have one copy of that particular chromosome. That means that the two options are only G or C. Now, what I do with this, I carry out what we call association studies. So I look into these positions in the DNA where we know that there is more than one option, and I do that for a large number of individuals. These days, we are working with about 100,000 participants. And the idea behind this is to see whether one of the alleles is, the frequency of one of the alleles differs really substantially when we compare its frequency in a group of individuals with a particular disease and the frequency on a group of healthy individuals. The main issue here is that maybe just random, like by chance, if I allocate these individuals to different groups, sometimes I might observe that there is a difference. So the idea is that you use some mathematical modeling to see that this difference in the frequency is larger than what we will expect just by chance. And in this case, I'm showing just one polymorphism, but in reality, we are working with 15 million, around these days, around 50 million variants, 50 million polymorphisms. So this is the typical way we represent these results. So this is what we call Manhattan plots. So you have each one of these dots will represent the result from a particular polymorphism. And in the x-axis, it will be represented the position, the physical position within the chromosome. So on this side, you will have the polymorphisms that are located at this end of the chromosome, and on the other side, the other end, and that from chromosome 1 to chromosome 22. And then on the vertical axis is a representation of how much the frequency between the two groups departs just by chance, right? It's a measure of that. Basically, I see this as a kind of a scan through the genome that gives you a clue of whereabout in the genome you need to have a look in more detail. So for instance, this, what we call association signals or peaks, will point me to, okay, let's have a look more in detail what's happening there. So let's do that. And this is a real example. So here you have what we call a regional Manhattan plot. Again, each one of the dots is the result from a particular polymorphism. And in this case, this is how we name them. They have these RSIDs. So now you can see that this particular polymorphism in this chromosome is located just over a gene, which is called ADCY7. So luckily for us, this is fairly easy for us to interpret. So when the variants, these polymorphisms are located within the genes, within the parts that will build the protein, we already know which is the code to the cipher. So we can use this, what's called Codon Wheel, and we can translate that into the protein. So if we start from here, from a group of 3 letters, which is what they call a codon, ATC, if you go here to the wheel, ATC will, ATG, sorry, will code for an M, which is a methionine. Now, if we continue, ASC will code for an asparagine, which is another amino acid, which the amino acids are the bricks within the protein. And then we reach the polymorphic position where there are 2 options. You can have TGT or TGA. If you have TGT, we'll code for a cysteine, whereas if you have a TGA, it will code for a stop codon. So this is really easy to link these to the functional consequence, because now you can think that the protein here will have the full length, it will continue, whereas in this case, we will end up with a protein much more shorter, so it's much more likely to have a biological repercussion. But unfortunately for us, this is only the case in 10 to 20% of all the GWAS signals. More than 80% of the signals will fall outside the protein coding genes. So this links to a project called the ENCODE project that was finished in 2012 that help us to understand what's the functionality of those regions of the DNA that don't code for protein coding, for proteins. And this is the most of the human genome. Now, we go back to the biology. We know that the DNA forms the chromosomes, but it's being packed in a specific way. So the DNA folds surrounding a protein, it's called a histone, with what we call nucleosomes. And I like to think on this like a kind of way of keeping things safe. So you have the histones and the DNA that is surrounding the histones in the nucleosome. And it's a way to keep the DNA safe. Like when you have tools that you don't need, you put them in a cardboard box and you put it into the attic because you want to keep them safe. When the cell requires to use that particular specific region of the genome because it needs the protein that is coded there, it opens and then the DNA is accessible, and that will allow to the machinery, the cell machinery, to create the messenger RNA, and then from there generate the proteins. Now, outside the genes, what we learned from the ENCODE project is that there are some regions that we call regulatory regions, and are the ones that really finally are able to switch on or off the genes, and not only that, to say how much of the gene, how much is going to be converted into, copied into mRNA, and then so on into proteins. So we have like two different regions. One are the one we call proximal regulatory regions that just align nearby where the gene is, and we call them as well promoters. And then we have another ones that might be located farther away from the gene. We call them distal regulatory regions, and we have two kinds. One is, well, additional ones, but the simple ones is the enhancers, and other ones that are called silencers. So how this relates with the common variation? Oh, sorry, I'm missing another element here, which are specific proteins called transcription factors that will bind those regions, that just bind the DNA, and they are called transcription factors. Now, how this links with the common variation and the diseases? Another thing I need to explain here is that the DNA is not a rigid piece of wire. You need to think about this like more of a kind of a string, where the DNA can come with themselves, and two regions that are really far away can't fold, and therefore this enhancer that was far away can be now perfectly close to the promoter region, and down. By doing that, what it will do is will tell the gene, I need to make many copies of you, right? Whereas if instead of an enhancer there is a silencer, what it will tell the gene is, I don't need copies of you, I do have enough, that's it. Now going back to the variants, what we observe now these days is that what we have learned is that our variants associated with the disease, when they are not located over the genes, they are specifically located over these regulatory regions, not randomly across the genome. No, they are over these regulatory regions. Now, this specific conformation might be true for a specific kind of cell. And in this case, I'm introducing our friend, the T cells, which are part of the immune system. And these days have become really famous because they are believed to be behind the immunity we get against COVID. So this conformation, the fact that this part of the DNA is open, this gene is being transcribed, and this enhancer is active, might be true for the T cells. But maybe the lung cells, that's not true. In the lung cells, that region is condensed. The chromatin is compacted because the lung cells don't need that protein. So it's condensed, it's packed. And then we have another cell where that region is open. This gene is not actively transcribed. We have another regulatory region here, which it's regulating, in fact, another gene here, and it's increasing its transcription. So, which is really nice is that, where do you think the variants that are associated with inflammatory bowel disease, which is an immune-mediated trait, are located? Well, they are in the regulatory regions in T cells, right? And what happens with the breast cancer risk variants? They are located in the active regulatory regions in breast cancer cells. So this is cell type specific. And what's open, what parts of DNA are open, and therefore which genes are being transcribed, is what makes the cells what they are, is what they gives their characteristic. Because at the end, the DNA is the same. All my cells will, in a normal cell, when it's healthy, will have the same amount, the same DNA, the same information, the cell of my skin or the same of my lungs. It's just how they use the information that will give them their characteristics. Okay, so now we can go to a specific example, and this is within breast cancer. So here we have results in chromosome 2 for one of the association signals linked to breast cancer. So we have this particular variant here that is located in an active regulatory region in breast cancer cells, and specifically it's located where one of these transcription factors I was talking before, one of these proteins binds, which is called FOXA1. And what is really nice is that FOXA1, in order to recognize where to bind, it can only do it when there is a T here, one of the alleles of this particular polymorphism. When there is a G, instead, it cannot recognize that region. And what it means is that when FOXA1 binds the T, a lot of copies of the cell produces many copies of this gene called IGF beta P5. Whereas with the G allele, that doesn't happen. There's still some copies being produced, but not in such large amount. So that's how in this particular region, we made the link between this particular polymorphism and that gene. Another cool example within the breast cancer context is that we realize that our associated variants tend to be located within the regulatory regions, but specifically where another transcription factor binds, where it's called GATA3. And what is really, really cool, at least I find it in that way, is that we have another variant that regulates how much GATA3 is being created. So now we have this nice network where we have some variants associated with breast cancer that regulate how much GATA3 is being created, how much protein. And then we have additional variants in the genome that regulate how much GATA3 is being linked to that region, and therefore regulate the expression of many other additional genes. So now you have this interconnection. And this happens not only for GATA3, but for other proteins or other transcription factors. So at the end, the final goal of these studies is for us to make a connection between that association signal, these signals we have here, and which is the likely target gene. Because you can imagine that when the association variants are located within the gene, for us it's really easy to make that connection. But when they are located in regulatory regions, sometimes we need to integrate many additional sources of information, and we need really intensive computational analysis in order to make that link. And once we have that, the story doesn't finish there. Then it's when my friends, my colleagues from the lab, who are molecular biologists, need to validate that. They need to go into the lab and design the assays that will allow us to really confirm that the two different polymorphisms are really acting on that particular gene, and they are modifying the expression, for instance, of that gene. And then they need to do another link, which is linking that gene to the disease. And that can take months or even years to confirm that, to validate that. So at this point, people, because we have been doing GWASs for more than 10 years, some people are now questioning why we keep doing it, why we keep publishing additional GWASs with more participants, where we identify more association signals and more genes. Well, because the more target genes we identify, the easier it is for us to see some patterns. And I'm going to show you one of the examples within the context of IBD in this case. So this Manhattan plot was published in 2010. And a couple of years later, within the International IBD Consortium, a larger GWAS was performed and additional genes were identified. And among them, there was one called ICAM1. Now, 2 years later, another paper from the International Bowel Disease Consortium, larger GWAS, additional genes, among them ITGAL. And a couple of years later, in a work that was coordinated by Carl Anderson's team, they identified, among others, again, 3 additional signals linked to these 3 genes, ITGAV, ATGA4, and ITGB8. And that was the point that they realized that all these genes in red are integrins. And I'm going to explain to you why integrins are relevant for inflammatory bowel disease. So integrins code for proteins that need to work together into what we call dimers. So in this case, ITGA4, which is, I hope you remember, it's one of the genes I was showing before, codes for a protein, ITGB7 codes for another one, and they together form what we call alpha 4 beta 7 dimer. This protein is located in the surface of the T cells. So it's a protein that is located in the surface of the let me go a little, on the surface of the T cells. The T cells are located in the blood, in the vessels, and the vessels, the walls are built by what we call endothelial cells. And some of these endothelial cells have some proteins in the surface. In this case, this one has one that's called MAdCAM1. So what happens is this T cell with that dimer goes in the blood and recognizes MAdCAM1 is the signal that the T cell requires for going away from the vessel and go to the tissue outside. But the important part here is where it happens. This happens in the gut. And the accumulation of these T cells with this specific marker in this protein in the surface is what is promotes the inflammation in the inflammatory bowel disease. So this is the specific link within inflammatory bowel disease. And this, what the important part is that this is happening specifically within the gut, within the vessels that are in the gut. So hopefully by this I'm able to show you that GWAS allow us to increase the understanding we have of the biology of this particular disease. In this case, it is IBD, but it happens the same with the other ones. But with that gain of knowledge, it comes something in consequence. So if we know which genes are involved in the disease, which proteins are involved in the disease, we might start thinking, well, I might target these proteins with a drug, and that might help us to treat the disease. And again, I'm going to show you an example, again, related with our friend alpha 4 beta 7 dimer. Specifically, for IBD, there are now a new group of drugs like vedolizumab and etrolizumab that specifically target that protein. So what it makes, it's canceling, it's avoiding the T cells to go to the gut, right? And then it's removing this pro-inflammatory action. So this is an actual treatment that is currently being used for IBD. Another example is what we call anti-TNF. Maybe you didn't see, but TNF is another of our target genes from the GWAS from IBD. So there are another group of drugs called anti-TNFs that specifically target that protein, TNF, and which is really producing really high levels within the patients with inflammatory bowel disease. So, but what else can we do with the results from the genome-wide association studies? We can learn a bit about the biology. We can start thinking about potential drug targets. But another result we get from the genome-wide association studies is as estimate of the risk. We get also how much risk is being conferred by each one of these variants. The thing is, in this case, or also in breast cancer, as I'm explaining in this case, are what we call complex diseases, where it's not only the effect of your genetic background, it's also the effect of other environmental factors. So each one of the variants I'm showing today will confer a really small increase in the risk, but it's the sum of the increased risk conferred by all these genetic variants together with the environment that can put you in a more likely position of developing that disease. This is true for breast cancer overall or for IBD. So one way of using that risk estimates is in what we call polygenic risk scores, and you might have heard about that. So I'm going to give a brief explanation of how we use that. So imagine we have a hypothetical disease where we identify 3 polymorphisms that are linked to the disease. And then we have the alleles for each one of these polymorphisms. For instance, for polymorphism A are uppercase A, lowercase a. For B is uppercase B and lowercase b, and so on. And now we have the risk estimates, hypothetically. For the capital A is 0.5, for the capital B is 0.3, for the capital C is 0.2. And now we have 3 people, and we have the genotypes. We have the specific information at each one of these polymorphisms. So this person has a capital A, because remember, we have 2 copies for each one of the chromosomes. So that means it has 2 times 0.5 capital A, 1 time 0.3 capital B, and 2 copies of C, 2 times 0.2. And if we sum this, will give us a polygenic risk score, or PRS, of 1.7. Now we have second person, we do the same, we get a PRS estimate for that particular person, and the same for the third person. Now, going back to a real example, we estimate these values. So for each one of the persons, we have 1 dot here in this plot, in this distribution. And this is a real example built with 147 polymorphisms associated with Crohn's disease, which is one of the major subtypes of inflammatory bowel disease. And this is what we observe within a group of people who has Crohn's disease. So you can imagine that with 147 independent variants, it's really the likelihood is not very likely to have to carry all of the risk variants. Therefore, there will be not too many people located here on this side of the distribution. In the same way, with people not having any of the risk alleles, that's really infrequent. So the majority of the people will be located in between here, right? Now, let's do the same with a group of healthy individuals. So this, you can see now that the distribution within the healthy individuals tends to be lower. Okay, now let's say we have another person. Now we don't know if it's a Crohn's disease patient or is a healthy individual, and we estimate with the same variance, which is the PRS. And now we get a PRS 0.01. Just because the 2 distributions overlap so much, just by doing this, we cannot really infer whether it's going to be a Crohn's disease patient or a healthy individual, just because of the distribution. So what can we do? How can we use this PRS then? And this comes from a work done in the team I was used to work before within the University of Cambridge, where they generate a PRS, in this case for breast cancer, and they were using 313 variants, as polymorphisms associated with breast cancer. Now we have a similar distribution, and instead of looking individual by individual to their specific PRS, we can now think on compiling the people into risk groups. So people within the 1% of lower distribution, 1 to 5%, and so on, right? And now we have people defined in these bins. What we do know about breast cancer? So in a normal population, one of the major determinants of breast cancer is age. It's one of the major risk factors. And according to that, and according to the population risk estimates of women in the UK are being offered mammographic screen at age of 50, because it's when they pass a certain risk estimate, a risk threshold. And this, as I'm saying, is based on the population estimates of breast cancer risk. Now, you can think about the previous slide, and you can see that, well, there might be difference in the risk between woman to woman. The risk might not be the same, is what we were seeing also in the previous distribution. So how can we use that? Well, in fact, if now you use that PRS and you look at which is the risk for the woman who has most of the risk polymorphisms, those women might pass that threshold, which is what defines why we are having the mammography at 50 years old, at a very early age, at 30 years old. Whereas there will be women that they have the least of the risk polymorphisms that might not ever pass that risk threshold. So we can now start thinking that we might use this PRS in order to identify the groups of women that might be, for which might be beneficial to have a mammography screening at early ages. So that might be one of the uses. So hopefully by giving these examples, I can I've been able to show you that we can use these GWASs as a tool first to understand the biology of a disease. Second, we can help us to define if treatments for a particular disease. And third and last, what we really want is to design a more personalized medicine. Now, what's next, and which are the trendy topics now on this field? We have big issue these days. So the problem is that the majority of the GWASs we have come from data coming from European population. And if we really want to make this as a tool to be able to develop personalized medicine, we really need to increase the population diversity. So we need to engage with researchers from other countries, and we need to engage with participants from other populations. Also, these days, because we are now working with really large studies, we can start looking more in detail in what we call the phenotypes. So for instance, for IBD, we have two major forms, which is Crohn's disease and ulcerative colitis, but there might be another phenotypes that don't share the genetic background. So now we increasing our sample size, we will be able to start looking into this. So this is probably two of the main trends and main lines to follow these days. And finally, I want to highlight that these are studies that involve a large number of researchers. In this case are the people from the Strangeways Research Lab, where I was doing my first studies on breast cancer, working with Alison Dunning, and the group of Carl Anderson. And it's not only a large amount of researchers, but also large international consortium, and also a large number of participants that really don't mind to share all their genetic data with us. And hope you have enjoyed this talk, and I'm more than happy to take any questions you have. Thank you.
Wow. Laura, I think that was amazing. Actually, I got lost at some point in the sense, like I was writing down a question and then you were saying something that I think probably replied my question. I'm not sure, but I was like, oh my God, I can't really cope with all the information. So thank you very much. It's been great. I loved it. I loved it. I didn't know anything about these studies, to be honest, like in the sense that we know there's a lot of data and the genome project and all that, but I mean, in my case, I wasn't sure how all of these data are being combined and analyzed and all that. So I have a couple of questions, but I'm going to just give a couple of seconds to people to maybe unmute themselves if you want to go for questions yourself or write it down potentially. Otherwise, I'll start shooting my questions. So let's see if someone wants to go for it. Okay, can I start with the first one then? Okay, so I have so many. So the Manhattan plots that you were mentioning, you mentioned that then most of the variants that you've seen are in those regulatory regions, right? So,
yes, they are located in the non-coding genome. Yeah, yes, not specifically above the particular
gene. In like a proper protein
coding gene, unfortunately for us. Yeah.
The thing is, so are there many of those that we still haven't really got to know what they do, like not even being able to pin down that is a regulatory function?
Yes, so we are, unfortunately, to make the connection, so when they don't fall in the gene, to make the connection between that variant and the gene, we need to use information from what we call expression assays. We need to see which are the genes that are being expressed in a particular cell, and then we can start making those connections. The problem is that we don't always have the data for the specific cell in large, large studies with enough data with the cell type we are really need to look into. So it's data that has been generated, and it will, it's a big thing right now to make that connection, and yeah.
So I guess exactly. Just let's imagine you find you're studying a particular one of those dots or polymorphisms.
Yes.
And then you say, okay, now I need to find if this has been already found to be a regulatory area or a gene. What do you look for that? I guess you have like a...
So did you see what, for instance, the ENCODE project was great because all the information they generated and was the first release was in 2012. And now they are keep updating that, and they are keep dating data for more different cell types. I will look into there. So you can have, they will have defined which part of the genome is an active regulatory regions in T cells, which part of the genome is an active regulatory, and so on. So by integrating that data from together with our GWAS, it's a starting point for making those links. But then you have another sources that can help you to do that.
So it's not that you're going to find it just in that.
Exactly. So you need to integrate data from other different studies to do that. And yes, to answer to your question, for instance, for IBD, we have right now 240 signals associated with IBD, and we keep working on that. We keep working on linking those signals to the gene because we don't have the answer for all of them.
Fair enough.
So big bottleneck on that part.
I see. No, it must be a bit chaotic, I guess. I hope it's getting a bit more under control because, I see these days people just trying to work like on how to get better data sets, or at least, how to make them available to the whole research community. So I guess it's really difficult to organize. I have a couple more, but I think we have two people who wants to ask things. So Mena, if you want to mute, just feel free. Otherwise, I can go for it then. So Laura, can you tell us how increasing sample size detects more variants of genes? Are those the more rare ones? And if they are rare, doesn't that mean they are less likely to cause the disease in most people?
You have, so this is, in stats, is what we call having enough power to detect that signal. When I was talking before about our ability to discriminate whether that difference in the frequency is more than what we expect by chance, is related with that. So that can be due to two things. One is that the effect conferred for that variant is really small. So we are starting to identify variants that have a smaller increase in risk than the ones we have already identified. And that's why we need more samples to be able to detect that. Or it can be that what Mena was saying, that the variants are becoming less and less frequent, and therefore we need a larger sample size to perfectly define which is the risk of conferred for that variant. So it's just what we call a matter of power, our ability to discriminate whether this is real or not. And yes.
Quick question about polymorphism and variants. When do you consider a polymorphism a mutation? Okay, this is a random delimitation that sometimes you start calling, you call it polymorphism when it's above 0.1%, depends, or sometimes you call it rare variants when it go below 0.1. But I come from a diagnostic lab. So for me, mutation has the implication of have a consequence effects. So I wouldn't call them mutations until we have really linked the effect with the disease. And sometimes it's linked to variants which confer much more larger risk than the ones we are calling here, we are talking today. So that's common variation. So for me, mutation would be
a variant that, for instance, disrupts BRCA1, and therefore just by having that variant, your risk of developing breast cancer is really, really high. So that's another field I haven't been touching today in this talk, but yeah, it's going to the rare variation spectrum, and just by having one, your chances are really high.
Okay, so yeah, I see that the complexity of linking all those data increases with that too. Okay, so okay, Leticia, if you want to unmute.
Hello.
Yeah, go for it.
Okay.
Move in the lab.
Yeah, sorry. Hello. I thought the talk was super great. It was super well explained. I was just curious in general, if you know how we can study rare disease, or if those kind of studies, like somehow they can be used for rare disease, like disease like we find nothing or not at all. Like I don't know if I'm clear.
Okay, for rare diseases, usually so now we have this spectrum again. It goes from rare to common diseases. Rare diseases, usually we link them what we call the mutations I was talking before, variants that increase very much your risk of developing the disease. And then by having one, they are disrupting a particular protein, you have very high chances of having the disease. Now, what we are starting to know is that this common variation might also act as modifiers of these mutations. And this is something there's a really cool group in Strange Ways, we are working with that. They're working with breast cancer patients with familial aggregation that have mutations in BRCA1 or BRCA2. And then they are studying how these common variants might tune the risk conferred by the risk variant. Let me know if that's, okay. So it's the combination. So the risk variant by itself increases a lot your risk of developing the disease, and then the common variants might be increasing a little bit. So it might be behind why, well, yeah. Yes, I hope this is.
I imagine it's a lot of statistics.
Yeah, I see a room for common variation being involved, but this is, much small increased risk.
And I was just wondering also if you know, if you know also about those kind of studies and what they have found for COVID, and if you have heard about it, because I've heard that those kind of studies were done for patients with COVID, and then if you know what we know from those studies, or
I guess I guess it feels very tempting to try and find something with COVID these days. And I guess it's short. It's a very short time, but maybe there's something. Yeah.
Yes, they are. There is a large. It's amazing how the people in the field has joined efforts worldwide. So in this field, GWASs and how this common variation might increase your risk of being infected by COVID or not. There are some efforts, some international efforts for doing that. And they have already found, again, some regions in the genome that might be linked to risk. But again, as before I was saying, these would be really small increases in risk of.
Thank you.
Following on that, can you retrospectively get those, like the data of people that you've studied for all the things and just try now for COVID-19? Because I guess it's more about coordination of data.
I guess you need to contact these people sometimes and get their agreement to participate on that. But yes, this is data that is there and is exactly.
You can just always come back and try to find
a link.
Another question, Emma, I don't know if you want to mute. Maybe I can go for it then. Has there been any research which in terms of IBD, has there been any research which investigate the effect of a specific polymorphisms at the drug-targeted genes on therapeutic response?
Oh, yes. I haven't touched that, but there is some amazing work from one of my colleagues, Alex. So when I was talking about the anti-TNF before, there are really new drugs. And what we have found is that sometimes let me go back. So these drugs are usually given after another treatment has been not been useful. They don't respond to another treatment because there is really, really expensive treatment. So, but the problem is that when we give that treatment, some people respond at the very beginning and then start responding to anti-TNFs. So what Alex managed to do is to find a region in the genome that is linked to whether you end up responding after, having a continuous response to that treatment or not. And it's at the end related with how we respond against those anti-TNFs and how we clear them. It's really, really cool how So he managed to find, again, a region in the genome that is linked to whether the participants can respond to that gene, to that particular treatment or not. So yeah. Very good question.
Great. Thank you for Emma and for all of us, of course. More things, because let's see if people go for more or not, because I was going to ask some more things, especially, I'm going to just make the most of the recently awarded Nobel Prize to CRISPR. And asking things like, well, no, it's really CRISPR related, but just imagine that you have, again, as you said, that you have two sets of that gene or regulatory area and all that. So can you silence somehow one of them and just if the other one is the good one?
Yes, so there is many ways of how CRISPR is being used, and I'm not an expert. My colleagues at the lab, they're amazing doing that, and they're actually using CRISPR these days. So you can use to silence a particular gene. You can use it now, starting using to specifically you have a cell, and then you can introduce a particular variant, and then the other one introduce the other one. So in that way you have, let's say, the same cell, and you can follow it, and you can see how the gene responds to having that particular. So there is a lot of things you can do now these days with CRISPR. You can run CRISPR screenings, which is what they are currently been doing with the results from our breast cancer fine mapping study, where we have, we end up having some variants that can go into the lab, and they were trying to modify these particular variants in cells to see how the genes respond overall. And it's really, really cool what these days they can do with CRISPR. Yeah, it's still, again, it's not easy at all. When my colleagues at work explain to me how much it takes, to transform a cell using CRISPR, and it's not just that I want to change that particular position and I'm going to know it. It takes a lot of work, and sometimes it doesn't work with some polymorphisms, and sometimes the cells that don't like that, and they decide to die because they don't like that change. So really cool, really useful, but yeah, as everything is not as simple as it seems.
Yeah, okay, yeah, no, and I guess, again, if you manage to change a couple of cells, then you need to, an adult organism, you would need to change all of them, right? I guess if you wanted to reduce the risk to a complete minimum.
Well, that's another thing, more little bit complicated, and which cells you change, because you don't want to change the cells that are going to be germinal cells. That will mean that you will transfer that to the descendants. So it's a little bit more tricky. Yeah,
exactly.
So ethically,
a long way to go, but at least, okay. At the lab level, there are things that, perfect. And also, this may not link at all with this, but I was going to say epigenetics, because you were mentioning things like, actually, I find it really interesting things like, increased risk of breast cancer with age and things like that. And, I don't know whether that means the tissue changes, and I guess certain changes may trigger more copies of the protein, that I don't know. But I guess, it's epigenetics also playing a role there, and how can you study that really or not, or you can't really study?
Yeah, it's some kind of related, because when I was telling you before that the region is open or closed, we do know that thanks to epigenetics, some epigenetic marks. So now we know that, for instance, remember when we were talking about the histones, there is a mark in a specific part of, for instance, it's called, I'm going to do the technical name, acetylation of lysine 27 or histone 3. That mark is an epigenetic mark and is what we correlate with active enhancers. So that's why how we these days we know that enhancer is. So that's a specific epigenetic mark, and that, as you can see from cell to cell, is going to change. So the DNA, yeah.
Okay, so you can see that.
Yeah,
the kind of specific PR and sequence and the standard sequencing. Because okay, I didn't know that.
Yes. Yeah, that's the mark that they use to define those regions.
Okay, because I wasn't sure whether that was something that could potentially change when you are sequencing the chromosome or something like, I don't know whether it's something.
You use sequencing, but in a different way, because you don't see it when you get the sequence for the DNA, you just get the letter A, T, C, G, right?
Exactly.
Yeah, but for instance, for epigenetics, you can use how it is. You can use a specific proteins that bind where that epigenetic mark is, and then you can sequence what is in that part. So in that way, you get the sequence just for the part where the protein binds. And that's how you know that that region has that epigenetic mark.
Okay.
But it uses sequencing, but in a different way, yeah.
Okay, fair enough. Yeah, because I was trying to get my head around how do they know, in terms of epigenetics, what can be on or off, which, okay, fair enough. So a bit more complex. Why not? Let's increase complexity. But okay, so Jessica, if you want to unmute again, a couple of seconds. No? Okay, so can a polymorphic variant have multiple effects, like increasing the risk of one disease whilst decreasing the risk to another? Which actually I have a question like, have you seen benefits to some of these polymorphisms too?
Okay, really good question. Within the what's next, I didn't intentionally include what we call pleiotropy, which is now something we are starting to see more in detail, thanks to initiatives like UK Biobank, where you have data about the disease status for many diseases, and then you have the genotype data for all of these individuals. And now you can start making the relationship that she was saying, where you can find a variant that is associated with the risk of two diseases, and this is really frequent. For instance, variants that increase the risk of an inflammatory disease or immune-mediated like IBD might also increase the risk of other inflammatory diseases like rheumatoid arthritis or another examples. So there is a big overlap on these regions. There might be also the case, I cannot give you right now the example of a risk of a variant of a polymorphism that increase the risk of one disease and decrease the risk of another one. And it's highly likely that that is happening as well. And this is another really cool trend in the future to look into.
But I guess it's easier somehow to identify what is unhealthy than what is healthy. So I guess a healthy person is just a healthy pet. I'm not sure how you can say.
So you can do that with the UK Biobank. So for instance, you have large, like 500,000 people that have been genotyped. So you have the genotype data for all of them, and then you follow them. So some people might end up being an inflammatory bowel disease, patient. And other people might be cancer patient, but now you can start studying all this because you have all the information. You have all the what we call phenotype data for all of them. You are not only measuring whether it's an IBD patient or not. You can start.
You can check in the future.
Yes, exactly, and you follow.
Fair enough. And I was going to, actually I was going to ask very briefly. This sort of like when you talk about volunteers, I'm not sure whether that's easily accessible. Like I don't know whether people tend to hear about these studies, or it's more like, like in the medical world, you would know how to volunteer for these, or they target a specific groups of people. Again, as you were saying, now we need to target maybe people from other ethnical backgrounds. So I don't know how that is advertised somehow, but it's something that you tend not to hear. I don't know whether you know, or whether you don't really.
Yeah. To be fair, while I was with my PhD, I was more involved in the part of recruiting people. But that was mainly being done, as you were saying, within the hospitals, where in my case, I was working with prostate cancer patients that were going to the radiation oncology department. And there it was where they were offered the possibility of participating into the study. For the large prospective ones, right now, I don't know, but I can look a little bit. If you are more interested on this, I'm more than happy to look into this. So just email me and I can look.
I may do that.
Sometimes it's much more
interesting, kind of to you know, like the banks and how do they recruit people? Yeah. I guess of course there's like a huge screening somehow before getting a sample and do all the work to get all that information. So great. So I think, yeah, it's a bit past 7. So I think we may release you because it's been a very, very intense hour, I think. So now we have to go home and just try and, go home in my case. I hope most of you are at home, and try to soak all this information. So yeah, people just saying thank you from the chat. I hope Ella can save the chat, and I hope I didn't miss any questions. But yeah, so everyone is thanking you. So thank you, Laura, and thank you everyone for joining us. I hope to see you in the next one. Bye bye. See you soon.
Bye. Thank you, Christina.
Bye.